So now we're going to talk about translation, which is the production of the protein. In the last lecture you heard me discuss transcription. We produce that messenger RNA molecule, it enters the cytoplasm, and now we're going to produce the protein. So this occurs on ribosomes, so either within the cytoplasm or in the rough ER on those ribosomes. Things that we're going to need, we need that messenger RNA molecule, we need ribosomes, we need transfer RNA, so ribosomes are the ribosomal RNA, just so you know. And then we need amino acids that we can use to produce the protein. And when we are producing the messenger, or when we are producing the protein, we read messenger RNA 3 nucleotides at a time. 3 nucleotides make up 1 codon. And we have a total of 4 nucleotides. So with that information, can you figure out how many codons there are? You take 4 times 4 times 4, or 4 to the 3rd power. You get 64 different codes. 61 code for amino acids, and 3 are stop codons. So there's a total of 64 codons, but only 61 code for amino acids. The start codon is AUG, or methionine. It codes, that starts all protein synthesis. That starts protein synthesis within bacterial cells. It starts protein synthesis within animal cells, and within human cells. We are animals, but non-human animals versus human animals. That makes sense. Insects, everything. AUG, methionine, starts all protein synthesis. And then there are 3 codes that stop protein synthesis. Translation, again, has that same 3 steps: initiation, elongation, and termination. And with translation, you can actually translate the same messenger RNA multiple times, and you can translate them multiple times in a row. Like you can have multiple ribosomes writing on the same transcript. So you can make lots of copies of the same protein. For those of you who've never seen a codon table, this is a codon table. A codon table contains the 3 letters for every single code, or for every single amino acid. And so in RNA, we have uracil, cytosine, adenine, and guanine. Here we have uracil, cytosine, adenine, and guanine. And then the same down here at each section. So we read our codon table. The first letter is going to be found in this first column. The second letter is going to be found across the top row. And then the third letter will be found within each of the rows one time. So UCAG for that section, UCAG for this section. So say you're looking for methionine, and you know the codon is AUG. First thing you do is you look for that A, so A is here, so you know that it's going to be somewhere in this row. We then go to the second letter, AUG, so we know it's U, so we go down here. So we know now methionine is going to be found in this section. To find the last letter, we go over here, and we go UCAG, G, methionine is AUG, AUG, methionine. That's as easy as it gets, reading these tables. You don't have to memorize a codon table. I will likely give you the codon table if I have, not if, I probably will have a couple questions. I'll give you the codons that you need, so you don't have to worry about that. So here we have our DNA strand, and then you have the DNA template strand is here. That means this is the coding strand, so we're using this strand to make our messenger RNA. So because T binds to A binds to U, C binds to G. If you look at this, it's going to be identical to this strand here, except for anywhere we have our T's, we change them to U's. So once we have our transcript, we now can produce the, we can then translate it on that ribosome. AUG codes for methionine. CCG, if we go back, CCG, here's C, C, and there's G, so that's proline. And if you look, they have proline written here. So if you go along this, you'll find all of the different groupings, all of the different codons code for these different amino acids. So this occurs on the ribosome. A ribosome is composed of a large subunit and a small subunit. There are 3 different sites within a ribosome. We have the E site, which is the exit site, and then the P site and the A site. I typically only talk about the P and the A site because the E site is just the exit site. So once the ribosome binds to the transcript or the messenger RNA, you have the first site is going to be where your first codon is going to be found, and the second site is going to be where the second codon is found. So you end up having transfer RNAs come in, and they transfer, they contain what we call the anticodon, which is opposite the codon, and then they hold the amino acid that is coded for in the transcript. So A binds to U, so we have UUU, AAA, UUU codes for phenylalanine, so that's going to go here. Once these two are bound in the P and A site, then these two amino acids form a peptide bond. And both of the peptide bonds, and these two peptide bonded molecules, are going to stick together. And then the entire ribosome moves. The first transfer RNA is going to leave to go pick up another amino acid. The second transfer RNA, it gets moved into the first site, and then we end up with another transfer RNA coming in with its amino acid and the anticodon. And we form another peptide bond between phenylalanine and arginine. This is going to occur all the way down. So this is my, again, this is something I made for my micro students, but it's something that I like and I think it makes sense. So this yellow structure, this is my ribosomal RNA. These little pink things are my transfer RNAs, and then this long green thing is my messenger RNA. And you can see here I have AUG, that's the code for methionine. So the first amino acid that is going to be carried is methionine. There's the anticodon carried on that transfer RNA. Second one is a transfer RNA carrying the anticodon. CCG binds here to the GGC, which codes for glycine. And the first transfer RNA moves, and the entire ribosome moves, and you have another amino acid coming in, and this is going to continue until we hit that stop code. Once we hit the stop code, we then the ribosome will come apart, and the transfer RNAs can go and pick up more amino acids. This polypeptide is now going to be able to fold up into a functional protein, and the messenger RNA can be retranslated if we want to. So if you didn't understand protein synthesis from my explanation, there's an excellent video from the Amoeba Sisters. It's about like 8 or 9 minutes long, so I'm not going to play it in this video because that would be a waste. But all you have to do is click on this link up here, be attached to the internet, click on this link, and you'll get to the Amoeba Sisters animation on protein synthesis. Also, if you're still struggling, come to class and ask, because the more questions you bring, the better our discussion portion is going to be. So here you have your DNA. DNA is transcribed into messenger RNA. This all occurs in the nucleus. It's then going to be processed, and then you move it out into the cytoplasm onto those ribosomes where we have translation occurring, and that's what this figure shows. The last thing we're going to talk about in this chapter, well, second to last thing, I guess, is cell division. So I know this is a short lecture here. It's only a 10-minute one, but I'm going to stop here so I can get my cell cycle in with the last, or in one video on its own.